My name is Cullen Buie. I'm on the faculty of mechanical engineering as an associate professor here at MIT. My expertise is in fluid mechanics, specifically microfluidics. My group studies how small particles flow through channels or how fluids flow through small channels, with applications in a wide array of arenas, from material science through biotechnology. Though recently my group has focused mainly on biotech, you might say, how we can manipulate small volumes of fluid in order to answer questions in microbiology and for cell engineering. In the case of microbiology and cell engineering, it's pretty interesting. I was at a conference about a decade ago, and while I was there, it was not in my field. It was in the area of synthetic biology, but it was an area I was curious about. I spoke to someone and they asked me, they were a program manager at DARPA, and they asked me the question if I had ever considered working on the problem of getting genetic material into cells. At the time, the government, particularly the military, was putting a lot of funding into figuring out how you can re-engineer cells in order to get them to do new things. So how do you get a cell and essentially hijack its machinery to produce new chemicals or have new functionality? This was an area I was interested in but didn't know much about, and after hearing about the problem was effectively you have a cell, and you could think of a cell like a computer, or you might even think of it, like your phone. So you have a phone, which is hardware, and what people were trying to do is upload new software or genetic code into cells and get them to do new things. You can think of it like apps for your phone. The problem was the upload process was way too slow. And so you were limited in your ability to reprogram cells. And so my group set about trying to come up with new ways of putting genetic material into cells to give cells a broader range of functionality. So there are a lot of challenges in this area. So you could think broadly, what my group is trying to do is make the process of putting new DNA or RNA or effectively gene altering material into cells. We want to make that process more efficient. And we want to do it for a wider array of cells. So there are a lot of cells that are used. There are microbes such as yeast and bacteria that are used, but this is also done for mammalian cells. In some cases, people would like to engineer a mammalian cell in order to produce a protein or a drug that will be very complex. And getting a mammalian cell to produce that drug, it's more efficient than using standard laboratory chemistry. But in other cases, people actually want to engineer cells in order for those cells to be the drug themselves, where you actually change how the cell functions, and that cell goes into the body and is able to act like a boost, you might say, to the immune system. And so to the first part, the applications around microbes, yeast and bacteria are mainly in trying to get those cells to produce things for us. So how can we get cells to produce additives for food or for materials, raw materials? Imagine being able to get away from using fossil fuels as precursors for many materials, because you can actually use microbes or microorganisms to produce the precursors for those materials, which would have, of course, effects potentially on the environment and on our natural resources. So that's one big class of applications, is getting microorganisms to produce things of use to humanity. Another applications of those microorganisms is, in some cases, you want those organisms to produce drugs or pharmaceuticals. So that's another big application. And a third growing application is the microbiome. So the microbiome it's really the human microbiome in particular. It's all the living organisms that live in and on our body. So it's known that the number of microbes that live on a human, there are more microbial cells than human cells on the typical human. These microbes are thought of almost like an additional organ because of the effect they have on our body. They're on our skin, they're in our mouth, they're in our gut, they're all over us, and they have a strong effect on our health. And there are people that would like to engineer microbes in order to perturb them for healthcare applications. And so imagine you take a pill, and that pill contains microorganisms that then affect your health. And so that's another application for engineering of microbes. So that's one class, engineering microbes for those applications. Another, I would say, class of applications is where you want to engineer cells that are more human-like. And I would say the biggest and most exciting one that people are considering today is using those cells for altering the function of the immune system. So one application that has gained a lot of traction, and it even has a couple approvals by the FDA already, is where people would like to engineer your immune cells to fight your cancer. So imagine you go to the doctor or to the hospital, they draw your blood, they isolate a particular type of cell, and then they upload gene altering material into those cells, put those cells back in your body, and they find your cancer and eradicate it. So that's the dream, is really re-engineering your immune system to give it a bit of a boost. And once again, my group is working on ways of doing that re-engineering process. How do you upload that material into the cell in a way that the cells will live, because those cells are now the drug, and so you want a lot of them to live, and that they'll be effective in the therapeutic outcome that you would like in the end.